So, after cellular respiration, it's not respiration, cellular replication. We're going to talk a little bit about gene expression. So, as we talked a little bit about before, somatic cells and germline cells do not have the same function or appearance. However, through genetic study, we know that every single cell in a particular organism has or contains the exact same genetic code. And unfortunately, not all of that genetic code is going to be used by that cell because it has a certain structure that it needs to do. And to do that structure and function, they have to produce unique proteins. So this process of protein creation is done through protein synthesis, which are broken up into two steps: translation and transcription. So those two together is the process that our cells use to turn the information encoded by our DNA into a usable form. For example, proteins and other functional molecules. Underneath the QCEE syllabus, we've got two important definitions that you guys need to know. I've copied these two directly from the QCEE glossary. And a gene will have regions of DNA that are made up of nucleotides. They are known as the molecular unit of heredity. And a genome is all of the genetic material in the chromosomes of an organism, including its genes and DNA sequences. These two I strongly recommend you memorize, along with any of the other definitions in the syllabus. They can come up as either short answer questions or multiple choice questions I have seen. So, within our DNA, I did touch on this a little bit earlier, but I'm going to go into detail now. We've got coding regions that actually code for a particular protein or are used as areas of DNA that create functional gene products. These are this actually makes up a very, very small part of the DNA. And you call these coding regions exons. So these are actually the ones that are useful in terms of creating proteins or gene products that end up doing a particular function for a cell. Every other aspect of the DNA is enclosed in a non-coding region. So these are regions of DNA that don't create proteins, but are responsible for creating other structures like centromeres, telomeres, introns, which is the general term for non-coding regions, and important sections of regulatory genetic material, which are sections of DNA that we don't really understand the function of yet. We know that some of them form the centers of the chromosomes, some of them form the telomeres of the chromosomes, but in total, a lot of the functions of introns are not understood by humans right now. The most important function that we do know that they perform is the regulation of genetic material, and some of them can also exist as ribosomal RNA and transfer RNA as well. So after just before going into the actual process of DNA, we need to understand how DNA actually codes for proteins. So before I mentioned that each of the strands are organized in a series of base pairs in terms of thymine, adenine, cytosine, and guanine. However, in protein synthesis, we want to be talking about this U base instead of thymine, which is uracil. Cytosine, adenine, and guanine stay the exact same, fortunately. And each of these groups of DNA in the exons, remember the exons are the ones that are actually coding for proteins, will be read in groups of 3. So each group of 3 can be read in the protein synthesis process, and they produce one corresponding amino acid. And you'll be given this table in any exam questions relating to protein synthesis if you need to get the amino acid chain that is produced at the end of the process using this particular table right here. So I'm not going to dwell too much on this, but this is something that you guys will need to get acquainted to quite quickly. Alrighty, now we're actually going to go on to the process of protein synthesis. So I'm going to try and explain this the best that I can. Just get my pen. And... Radio. So we've got our first step in protein synthesis, which is transcription. Now, the reason why this is called transcription is that this process stays in the same language. It stays in the language of nucleotide base pairs, language of nitrogenous bases. Oh, sorry. Bases. It occurs in the location where DNA is. So this is the nucleus of the cell. And essentially, the gene that needs to be encoded will be exposed. So for example, if this is the particular gene that we need to encode, it will be exposed and copied by an enzyme called RNA polymerase. RNA polymerase will get in here and copy the enzyme by attaching complementary base pairs. However, the important thing to note here is that instead of finding adenine from the original copy of DNA will bind to uracil in this strand. Now, this forms a single-stranded molecule of DNA, which is known as RNA, ribonucleic acid. Specifically, this happens because RNA has the ability to leave the nucleus. And that allows it to reach all of the structures that actually end up doing protein synthesis later on. So RNA has the ability to reach the nucleus. And this specific strand of RNA is called mRNA or messenger RNA. Think of it as containing a message that needs to get processed into a protein. However, fresh off the DNA transcription, this is still pre-mRNA because it hasn't been processed properly, but that'll happen in the next step. After transcription occurs, the mRNA molecule moves out of the nucleus into the cytoplasm for the next step. Moves out of the nucleus into cytoplasm. Okay. Then what happens is a series of steps called gene splicing. So this is what is called a post-strand transcription modification. If you're following along with the syllabus, they use this term, post-transcriptional modification. But basically, you end up with the original strand of pre-mRNA, which contains a mixture of both exons, which remember are our coding regions, and introns, which are our non-coding regions. Obviously, if you're trying to code a protein, you only want coding regions of DNA in there, otherwise you run the risk of mutation or the creation of a wrong protein. So gene splicing will occur where all of the introns are then cut out of the pre-mRNA strand and all of the exons are glued together, forming what is called a mature mRNA strand that is ready for protein synthesis and minimizes the risk of mutations occurring. After that, we end up in our final step, which is translation. So the mRNA strand is now brought into the cytoplasm of the cell, specifically in the area with ribosomes. To ribosome rich area. So we end up with the mRNA strand right here. And then as it As it moves into the cytoplasm, a ribosome will come into contact with it. Imagine this is a ribosome. And the mRNA strand is read in groups of 3 called codons. Codons by structures called tRNA. So tRNA are these weird looking structures that kind of look like keyholes, like this, that are anchored onto by the ribosome. And they pair up the mRNA strand with a thing called an anticodon. So, for example, if the mRNA original strand had a GAU code, the anticodon will pair it up with a CUA. The tRNA will then read the GAU codon and go, okay, GAU corresponds with, if I go back to the table from the start, I've got GAU ASP. So that's the amino acid that it needs to create. And then off the top of the tRNA, we will get an ASP amino acid. Then another tRNA molecule will move on to the next set of codons. So like so. So for example, if I had UAC, the anticodon will read AUG, and then UAC would form a TYR amino acid. So we still have our ASP. This orange one from before breaks off, hands the ASP to the next one, and then off the next anticodon will produce a TYR amino acid. This ribosome will then continue to form down the mRNA strand until it hits a stop codon. So if you look back onto the table from before, there are 3 stop codons, UAA, UAG, and UGA. You do not need to memorize this. If you are required a protein translation, you'll be given the table itself. But once the stop codon is hit, the ribosome and the tRNA will stop the translation process, and the chain of amino acids, which has been produced, also known as a polypeptide chain, the polypeptide chain will break off the tRNA and the ribosome. And condense into a 3D structure known as a protein. So essentially, this chain of amino acids will then bunch up into a 3D structure, kind of looks like a chicken nugget, and this is this is eventually the protein that ends up being produced. The reason why we call this translation is that we're going in the language of DNA, which is nitrogenous bases, into the language of proteins, amino acids. Because we're changing languages, it's translation. Language of amino acids. There we go. So that is the process. I hope that makes sense. And here's another structure of it right here. You can see this teal colored structure is the ribosome, and it anchors onto the mRNA strand, which is like the pinky peach one that you can see here. The golden keyhole looking structures are your tRNA molecules, which form an anticodon to the codons from the mRNA. After reading the anticodon, it produces this amino acid, which are connected together by peptide bonds. If it gets to the stop codon, which are your UAA, UAG, and UGAs, we detach the polypeptide and it condenses into a protein. Just one other thing, the protein translation process will only start once an AUG codon has been read. This is our start codon, the red one down here, which produces the MET amino acid. And that's how the ribosome knows that, oh, this is the starting process, and it'll go down the chain of the mRNA until it hits the stop codon. Um, so after that, um, oh, so after that, each of these codons will be read into an amino acid. So for example, AUG right here is our MET. I'm just going to translate it with you guys. The answer is on the next slide if you want to move forward, but I've got, oops. I have AUG, which is MET, ACG. So the way that you read it is you go down this line first. ACG, which is THR, UCA, which is serine, GAA, glue, AAA, lysine, AAU, ASN, CAA, GLN, CGG, ARG, GCA, ALA, GUG, Val, UCC, which is also serine, and then CCC, which is pro. So notice here we started off with a MET, so this is the start of the protein code. However, we didn't end up with any of the stop protein code. So this is only a fraction or part of a code for a protein. You'll only get a full protein code if it starts with MET and then ends with any of the stop codons. Like so. So here's the answer for it if you were watching the recording and you wanted to skip me going through it. But once again, this is only part of a code for a protein. In terms of what you would need to know, these are the syllabus dot points that I have transcribed over. So you need to know the difference between, or the definition of the term gene and genome. You need to understand that genes include coding exons and non-coding DNA, which have some functions like centromere formation, telomere formation, and introns, as well as functional RNA like tRNA, mRNA, and rRNA, which is ribosomal RNA. Recognize that many functions of non-coding DNA are actually yet to be determined. You will also need to know the process of protein synthesis in terms of transcription of a gene into messenger RNA in the nucleus, and then translation of mRNA into an amino acid sequence at the ribosome, and refer to transfer RNA codons and anticodons. So the way that I actually learned how to do this is I had like a whiteboard or a blank piece of paper, and I would draw out the process using diagrams, and every single time it would just go into my head much better than me trying to learn how to explain it using words. So